pleasure to start this action, which is going to last 4 years. So it's our first meeting. We're going to be about 70 today. Before introducing the action, I will give the floor to the Dean of the Faculty of Biology, who is welcoming us, and then I will introduce the day.
Their guests, their colleagues, it's my great honor to welcome you to this event, this first annual meeting of Cost Action Route of Benefits. It's my truly honor to have each of you here as we embark on this journey towards fostering sustainable agriculture through the exploring of root-associated microorganisms. Your presence underscores the significance of our collective efforts in advancing agriculture sustainability. And we are excited to embark on this collaboration ever together. I'm really glad that such events, the first annual meeting, is really taking place in person. Also, the COVID pandemic has taught us to quickly and effectively meet remotely on Zoom, Meet, or other platforms. Nothing can replace face-to-face meetings. And personal acquaintance with allows to better reveal the personality of each scientist so that cooperation will be successful. I'm pleased that the first annual meeting of this event is taking place right here at the University of Latvia in this House of Nature. This time for us at the University of Latvia is currently full of changes and challenges. External and internal consolidation of the university is currently undergoing. A new rector of the university was elected less than 1 month ago, and structural and management reorganization is undergoing at the university. On the one hand, it is a very turbulent time for us, but on the other hand, it's time of opportunity for all of us. We are very much hope that these ongoing changes will bring positive changes in the development of science, scientific funding, and cooperation, and that such events will become for us daily routine. On the other, in order for the event to take place, I would like to say thanks to all members of the working group who participated in the organization of this event. But I would like to give a special thanks to our colleague, senior researcher Olga Mutere, who is able to organize these things in very calm and obstructive way. I hope that today's weather, which is not very typical for Latvia at this time, today is snowing, that this weather will not be an obstacle to an intensive exchange of ideas, new ideas for the future scientific projects. And I wish you all to a creative time together. I wish you to appreciate the importance of your scientific results, and above all, new ideas for the future cooperation. Thank you.
Thank you. I would like to thank you again for welcoming us here, and also a special thanks to Olga and her team for organizing the meeting. Also to our grant manager, Yara, who have made the administrative organization, and the core group who has prepared the program of this meeting today. So what the objective of the meeting today is to get to know each other, to organize the work in the working groups, so that will be the session tomorrow morning in the different working groups, and also to discuss the future activities of the network in the next years. We can also have open discussion tomorrow morning. Actually, most of you, they have registered to different working groups, so you will have to make a choice for tomorrow morning, deciding which working group you can participate the most and go to this working group. So start to think about it to be ready for tomorrow morning when we are going to split in a working group. So I will just make a short reminding of the objective of our action. So the challenge we have identified when we wrote the proposal were that the community for researchers on root-associated microbes is fragmented by type of microorganism, nitrogen-fixing bacteria, mycorrhiza, plant growth-promoting bacteria, or fungi. And we thought we have the same objective for sustainable agriculture, and we thought it was useful to have all the community together to think about methodology and creating consortia. We also found that our topics are underrepresented in the European call, so we can make lobbying to have our topic more present in this call, and we are going to discuss this tomorrow as well. We think there are limitations in tools and methodologies for characterization of the microbial community and their function. There is also limited knowledge in the complex interaction, meaning in interaction between the different microbes and also with the environment, and that's a challenge for research. We also think that there is the potential for this microorganism to mitigate the effect of the climate change, which is not completely integrated in the research project, and that could be a research front. There is also potential for plant breeding to better benefit from this interaction that is not fully exploited. We also think that the scientific result or recommendation does not always reach the end user, and we can collectively make an effort for transferring our knowledge to the stakeholders. And we think also that the commercial inoculants does not always meet the expectation for long-term management of the microbial community in the soil, and there is also a research front on this aspect. So that's all the challenge we have identified. And we have proposed this network, which is composed of 4 working groups. They're going to be detailed this afternoon. Up to now, we have 336 participants. There are probably going to be slightly more in the next year, from 33 countries. 32 of these have MC member, and there are some additional participants from countries without MC member. So, and what are the activities we are organizing? So they are quite typical for cost action. They're going to be meeting and workshops. So one meeting, this one, and one workshop in September. I'm going to give more detail about this also later. There is a training school which is organized in June about methods for meta-analysis. There is already a registration open for this training school. It's going to be online to the meeting. There was already a first call for grants for short-term scientific mission. There's going to be a second one later. I'm also going to give more detail this afternoon about this. And we have also dissemination activities we plan to do during these 4 years with scientific review, policy reports, and we also plan to prepare teaching tools we can share all together. Actually, this slide will be available on the website, and you can use it also to make publicity for the networks when you go in conference or in different activities. Okay, so we also have some deliverables we need to produce during these 4 years. We are not going to read them, and they are all available also on the website. The only thing I want to point out, some of these, at least the 6 first deliverables, should be produced in the first 1.5 years. So we are now around 6 months after the starting. We still have 1 year to produce the first deliverables. So there are either publication, review of the literature of on the fields, or reports, or guidelines. But one of the goal of this meeting also is tomorrow morning in the working group session to organize different working groups to prepare these deliverables we have to produce. One of them is also a list of all the groups which are working in the field. So a survey was sent by the MC member to the people who are potentially interested in their countries, and we're going to have the reports now of the feedback from this survey in the countries. Maybe it's not yet fully exhaustive, but we had something like 230 answers to the survey. So we already have a good list of the community in your different countries. We can still improve it, and once this list is it's exhaustive enough, we are going to make it available for all participants, and this will help people to identify potential collaborators or place where people can think to go for STSM, for short-term mission exchange. And we hope this also going to help to create consortia if there are calls to which we can make application in the future. Okay, so the activities for 2024, I already said it, there was a first call for STSM. There's going to be a second one soon. So 2 meetings, this general meeting, and 1 workshop on non-academic stakeholder-driven topics. It's going to be in Portugal in September, and this will be dedicated to inoculants. So we hope to have in this meeting more private companies come in and discuss about application of inoculation. Training school have already introduced it. The survey inventory of the group laboratories are also introduced it. And soon also we are going to open a call for inclusiveness target country for grants to go in a conference. So this is only dedicated to ITC country and especially for young researchers to assist to a conference this year. So in terms of budgets, so the total budget is a bit more than 130,000 euro. So it's mainly dedicated to meetings. The 2 meetings, you have the details, so it's almost 100,000 euro for this. The training school, because it's online, it does not cut a loss this year. The ITC conference grant, they're going to be 5,000 euro dedicated to this. The short-term scientific mission, 26,000 euro. We have already spent half, and there is still half for the second call. And the last part is the dissemination and communication product, and it was mainly the website design. Okay, so the program today. This morning we're going to have the presentation by the MC member of the community in their countries. So we're going to have 2 sessions, a break between. Then we're going to have lunch, and this afternoon we're going to have a working group session with first an introduction of the objective of each working group by the working group leader, and then 3 presentations by different participants of the action. On the different working group, and then final 2 talks about communication and STSM. We're going to have dinner tonight in Riga Center, and tomorrow morning we have 2 sessions. One open discussion, 1.5 hours all together on different topics. So I ask you if there are topics you want to discuss. I received some email from some of you. If there are specific topics you want to discuss, it's still open. You can still send me an email for tomorrow morning. And then we're going to have a working group session organized by the working group leader. And then there's going to be the management committee meeting only for the management committee member tomorrow afternoon. So this morning and this afternoon, we'll have many presentations, not a lot of time for discussion and asking questions during the session, but we have many breaks. So keep your question for discussing with the people who are presenting during the breaks. If for the people who are online, if you want to communicate with the people who have presented, you can find the email on the website of COST. All participants are listed on the COST website with their email, so you can easily find their contact. And yeah, last point, if you still have ideas or things you want to discuss tomorrow morning, it's still open until tomorrow morning. There are also a number of posters just outside in front of the room. There are 15 posters, so there will be time during lunch and end of the afternoon to see the poster. And now we are going to start the presentation by the MC member of the research network in their home country. We are going to follow the alphabetic order. So I will call you one by one. First one is Albania.
Here.
Yeah, you can come here.
Please, to access Wi-Fi, please find in the list the LU Wi-Fi. Go there and please to put this login and password. It should work.
That's why it's yeah, but it's here.
Ladies and gentlemen, for the internet, you still didn't connect us. That is the LU Wi-Fi, and after you need to put the login which you have in the payment.
Here the Wi-Fi and the password.
Yeah, now it comes, but it's not sure. Sorry, duplicate screen. So we should find the duplication. That's it's okay. Thank you. Okay.
I will keep the time on, so like 5 minutes.
Yeah, for sure, yeah. Okay, hello everybody. I am Ardona Demirai from Agricultural University of Tirana, here representing the Department of Environmental and Natural Resources there, a lecture of environmental and pollution and remediation. So Agricultural University of Tirana is founded in 1951. The first university that takes care of agriculture and food safety. And we have 5 main faculties. Faculty of Agriculture, Biotechnology, Faculty of Economy and Agribusiness, Faculty of Forestry, Veterinary and Medicine, and we also have an Institute of Genetic Bank. At Agricultural University of Tirana, we have 23 regional programs and 1 integrated for second cycle, an integrated program, 19 Master of Science cycle, and also 20 professional Master Science, and 1 Executive Master, and 5 PhD programs for each faculty and each topics that they cover. On the other side, we have also ongoing projects, so 4 Horizon European project. 2 of them are launched this year. The FITO is the One Horizon project, and beaming on bioeconomy, where I am also part of this beaming project. Our staff is engaged on several cost actions, and me, for example, I am in 2 cost actions. In this cost, I am launched through only this year, so after 6 months difficulties with national contact points, but this came a positive answer. And we have also CIPUS project, Erasmus project, and more projects about financed from national agencies also that cover the research in master and PhD students engagement. In the other hand, we have as department the Laboratory of Soil and Water Qualities, but in the same time, for 2 years, we have established the COVID-19 laboratory, a new investment from a collaboration of GIZ in Germany and Agricultural University of Tirana, where we implement the early warning system for water quality evaluation of COVID-19. And in this laboratory, we are engaging to fulfill all the process of determination and preparedness of antimicrobial resistance. And in the other hand, we can use this laboratory to cover other topics that are engaged on a microbial field of study. The equipment are installed are very new technology, and the reason why I represent here is it to establish other methods for to wide the rate of laboratory analysis on a field of root benefits and microbiology of the roots. In the other hand, our team that we are engaged mainly on water and soil quality, but my colleague here, Nereida, is the one that is very engaged on phytomicrobiology, and the other working group that we work mainly on water and soil quality in our department are represented here, and they have published on peer review, and you can check if you want to collaborate for further projects or future collaboration. So that's my presentation. So I have kept time, hopefully.
Austria. No. Ah, yeah, okay. So, but you have another presentation. We will search it after. Okay, I make it after.
Good morning, everyone. My name is Hanna Koch. I'm representing Austria. I'm from the Austrian Institute of Technology, or short AIT. We are located in Tulln, which is a smaller city town next to Vienna. In addition, in Austria, we have currently in this cost action in total 4 research groups. So 2 of them are located in Tulln, 1 in Vienna, and 1 I haven't put in the presentation, but it's located here in Innsbruck at the University of Innsbruck. So what are the different institutions of Austria involved in this action? On the one hand, we have the University of Natural Resources and Life Sciences, or short in German BOKU. Here we have two different institutes. On the one hand, the Institute of Plant Protection, which is located in Tulln, and on the other hand, the Institute of Forest Ecology, which is located in Vienna. So the Institute of Plant Protection is represented by Karin Hage-Ahmed. And in this institute, they are mainly working on, as the main already mentioned, plant protection, phytopathology, biological control. They mainly work with Fusarium, Phytophthora as pathogens, but they also work with beneficial fungi and also bacteria like Bacillus. So that's the main lab beds, so to say. They, like I guess most of the institutes, they do have a variety of different tools, including molecular biology, classical microbiology, and they do this on different scales, like all of the institutes I'm representing now, so on the, for Austria, so in vitro, in climate chambers, glasshouse, but also field trials. The second institute of the University of Natural Resources and Life Sciences is the Institute for Forest Ecology, located in Vienna. So there Kati Retzer is represented, and they mainly work on the plant side, so mainly on plant physiology, root phenomics, and root development and things like that. So yeah, as you see, it's not my expertise, so sorry for that. But yeah, they do pretty awesome things, looking at how cells develop in terms that a root can grow. So a huge expertise in terms on the plant side on the one hand, and with the Institute of Plant Protection more on the biocontrol side. And then we have the Austrian Institute of Technology. So that's my research home, let's put it like that. We are the largest non-university research institute in Austria. We are also located in Tulln. In general, the AIT has a lot of different areas of expertise, and more than 1,500 employees. So not all of them are working in Tulln, obviously, but only our bioresource unit, which is led by Angela Sesic. We do have also a broad range of topics. So we have plant experts, we have fungi experts, we have bacterial experts. I mainly work with nitrogen cycling microorganisms and the plant microbiome in general, which also of course includes microbial fertilizer, and other experts are also working on the biocontrol field. And then the person I also quickly want to mention, but it's not in the presentation, is Nadine Präg from the Institute of Microbiology at the University of Innsbruck. So that was more on the left side of of Austria, so to say. So in the west. And she is mainly working on the roots and soil microbiome, so mainly doing omics techniques, with focusing on the methane metabolism in soil and rhizosphere. And what I also quickly would like to mention, very, very quickly, we do have, in Tulln, we do have a training course and workshop for microscopy and how to decipher plant-microbe interaction using different microscopy methods. So please check out the website, and if you have any questions for this, please come and talk to me. Thank you.
Okay, the next one is Belgium.
Good morning, my name is Bart Cotin, and I'm going to try to give you a quick overview of the survey that we sent around in Belgium. So we had 3 research groups contacted that did not respond, but 6 research groups from 5 institutions completed the survey. And then to make it simple, based on the research that they do, I took the liberty to roughly arrange them into 3 topics. So first topic is bioprotection against biotic stresses and abiotic stresses, and biostimulation of plant growth and plant defense. There are 3 labs that doing this kind of research. We have the lab of mycology of Professor Stéphane de Klerk, who is also an MC for Belgium. He's working also on AMF, and he offers an international training on in vitro cultivation of AMF. You can see the link over there. The second lab is the lab of plant health and protection of Professor Barbara de Keuning. That's in Leuven. They focus especially on Agrobacterium rhizogenes and do mechanistic analysis on plant immunity. And they also have a big project called SynBios, where they develop synergistic biocontrol techniques, and this is especially on strawberry. And the third group is the Rhizosphere group of Professor Sophie Hoormachtig from the Flemish Institute for Biotechnology. And they study especially the interaction of plant roots with neighboring organisms. They also have a focus on AMF, Bradyrhizobium, Streptomyces. And I saw in the program that there are 2 speakers from the Rhizosphere group, so this afternoon they can tell you more about the research that's happening there. I missed pressing the slides. Okay, for the second topic, we have two research groups. One is from the University of Ghent, from Professor Caroline de Tender, that's microbiology group, and the other one is the plant sciences unit of the Flanders Institute, Research Institute for Agriculture, Fisheries and Food. They have a close collaboration, in fact, on rhizosphere microbiota in relation to plant and soil health, and also the influence of soil additives like biochar or chitin on this rhizosphere microbiota, and even a European project where they collaborate on presence of microplastics, not in the sea, but here in agricultural soils, and eventually bacteria that can break down the microplastics, and at the same time can be PGPR or defense-stimulating bacteria. Last, third topic is, I called this topic antimicrobial technologies, more or less related to biocontrol. There we have the lab of microbial processes and interaction of Professor Philippe Jacques in Jean Blou. They have a focus on the secondary metabolites related to biopesticides, and they also study the mutualism between Bacillus philisensis and AMF. And then we have the lab of Professor Rob Lavigne in Leuven, lab of gene technology, and they do especially phage therapy in agriculture and bionic bacteria, developing bionic bacteria, means bacteria that are carrying virus genes or even virus particles. Okay, I think that's the quick overview. Within time?
Yeah, perfect.
Okay, thank you.
Next one is Croatia.
Good morning. I will present you research groups in Croatia dealing with beneficial microbes in soil. We are only 3 groups. One in Zagreb, one in Osijek, and one in Split. Colleagues from Zagreb, University of Zagreb, Faculty of Agriculture, Department of Microbiology, they deal mainly with nitrogen fixers, but lately they also deal with PGPR. I think they are the biggest group in Croatia. They deal with molecular biology, genetics, epigenetics. They isolate and identify these bacteria. They have a national collection of them, and they experiment mainly on legumes and Arabidopsis. The other group is from Osijek. We have colleague Jelena here. They are dealing mainly with pathogenic fungi, but also on beneficial endophytic fungi isolated from weeds and their interaction with wheat, maize, wine grapes, cherry. Also, it's a field of microbiology and phytopathology. And the third group, I come from this group. We are only a group of 4. Our biggest efforts are interactions of grapevine, virus, and arbuscular mycorrhizal fungi. Recently we had 2 national projects on this subject, and hosts that we are interested in are Mediterranean crops, mainly grapevine and olives, but also some aromatic herbs. And that's that.
This one is Czech.
So thank you. I'm presenting Czech Republic. My name is Martina Janoušková. As you can see, we are 9 research groups in 8 different institutions, most of it in or around Prague. But we have also 2 other big cities and big universities represented in Brno and in České Budějovice in the south of the country. So, I have grouped the institutions by the approaches, more or less. We have 3 institutions involved which really work in basic research. They are institutes of the Academy of Sciences, all of them in Prague or around. 2 of us, the group of Jan Janse and my group, we work with emphasis on Arbuscula mycorrhiza, but also with other, with different mycorrhizal types. We also use quite similar approaches, mainly in experimental work in greenhouse or growth chamber scale. The difference is maybe that Jan works a bit more in the physiological way, also applying isotopes is a big expertise of him. And we focus in my group more on communities, maybe on diversity of mycorrhizal fungi, on the relationship between diversity and functioning of mycorrhizas. Then there is the Institute of Experimental Botany, where we have Sándor Forcek, whose expertise is in isotopes as well. And we have Tatiana Kalachova, who works more with plant pathogens. And luckily she will have a talk in the afternoon, so she can present herself much better than I could do it now. So, and then we have 4 groups or 4 institutions which are more involved in agronomic research, more in the applied field, and they are in Prague and in different locations. The Crop Research Institute, which belongs to the Ministry of Agriculture, is represented by Markéta Marečková, who works mainly with the rhizosphere microbial communities of crops, and they do a lot of field screenings and also field trials, and also have nice publications on these topics. The Agritech Institute is located in the north of the country, and they are really much more practitioners. They have a lot of contacts to farmers. They also do regular workshops for farmers. They made a lot of field trials, and their main expertise is on pea breeding, but they work also with other leguminous species. So they are really interesting partners, especially because they have many contacts and many established links also to the end users. The 2 universities which are involved, one is really Agriculture University in Brno, and the other one is the faculty of the University of Agriculture Faculty of University in South Bohemia. In the first one, they work with plant pathogens mainly, and the University of South Bohemia, Bojana, she works with soil bacteria, soil fungi, but I wasn't able to get much more details from her about the work, so maybe I hope we will be able to meet her at some of the next meetings. Yes, and then we have the Symbiom company also in the network. Maybe you have heard about it as well, represented by Aleš Láter. And this company is really focused on the production of inocula of mycorrhizal fungi. So, and if it should be a network, so this is just a small summary. You can see that at the mutualist side, we collaborate quite intensively. We know each other, but we don't know so much within our country the people focused on plant pathogens, so I'm happy we have them here as well in the cost action. Thank you.
Next presentation is for Panma.
Hi, good morning. My name is Marcela, and I'm representing the research groups in Denmark. And actually, I've just put the ones that actively say that they would like to either host researchers or be very involved in this. And hopefully, in the future, we can also involve the rest. We have the Aarhus University. That is where I'm based, in the Department of Molecular Biology and Genetics. And we are the plant molecular biology section. Actually, this section, we are around 60 people, and we focus in different areas, and that is what I'm going to say in the next slide. And then for the University of Copenhagen, we have the Department of Biology in the section of terrestrial ecology section and fungi ecology lab, and also the Department of Plant and Environmental Sciences, microbial ecology and biotechnology. Actually, all these groups, we focus in very similar things, and we collaborate already. So it was a little bit different to put it within the topics, because we have all the topics together, so I'm going to keep it as the university. In this part, in the plant molecular sections, especially in the area that I'm working with, we are focusing mostly in the legume rhizobial interaction, but we will also have a lot of synthetic communities and working with intercropping with other cereal crops. We go all the way from molecular techniques to the production of inoculants and testing not only in lab and greenhouses, but also we have field trials. We focus in rhizobial inoculants and I mentioned already a lot of intercropping. In the part of the University of Copenhagen, we have two groups, one from Associate Professor Mere, that they work more in the part of microbiology interactions, also inoculant, but all related with plant growth promotion, not just rhizobia. Isolation of novel bacteria, and bacterial association with fungi. In the other side, with Professor Rasmus, also they work more focused in the fungal ecology, community ecology, root symbiosis, and mycorrhizal fungi. And the good thing is, actually, within our groups, we are already funded by several Danish funding bodies, especially Novo Nordisk Foundation, where we can have intergroups. So if you contact one of us, we are going to be working together. So if you have any more questions, please just contact me in the breaks.
Okay, next presentation is for Estonia.
Good morning. Very happy to present the Estonian root beneficial microorganism working people. Our answers to the form were fairly brief, so I'm presenting them, which doesn't mean that there wouldn't be more teams in Estonia. So I got answers from 2 institutions, 2+ research teams, both are in Tartu. So the first one is Tartu University, where I am from. So my name is Maarja Höpik, and I'm in the soil ecology team, working with arbuscular mycorrhizal fungi diversity, community ecology, biogeography, and so on. We also increasingly involved in agricultural aspects such as nature-based solutions of the microbial inoculants. In our university, there are also a number of other teams which are working in similar topics and could be considered for the STSMs or collaborations. So I have just broadly mentioned there are plant ecology teams there are mycology teams. The names would be Professor Leho Tedersoo or Professor Marina Sevchenko, Professor Mari Mura, and so on. So there would be a number of different aspects about AM fungal diversity, AM fungal interactions with natural plants or plants in agricultural systems, in artificial systems, at global scales, local scales, forests, grasslands, and so on. We are also quite intensive in the bioinformatics and all kinds of molecular aspects of describing the AMF communities. Then we have in Tartu also Estonian University of Life Sciences, and over there in the Department of Horticulture, Angela Kort, he's studying blueberry associating fungi. Angela is also here, she's sitting over there, and she has a poster, so you have a chance to go and chat with her about ericoid mycorrhiza, about the soft roots quality in relation to ericoid mycorrhizal inoculations and the production in the field and in the experimental systems. So that's the brief intro about Estonia. Thank you.
Next presentation is for France.
Okay. Thanks. I'm Florian Frugier, so I'm coming from the Paris-Saclay University, so that is nearby Paris. And that's one of the main places where there is groups interested in the cost action and in symbiosis. But there are also many other places in France that deal with these topics, and notably in Dijon and in Toulouse, from where Benoit is coming. So we have different institutions involved, mainly INRAE, that is dedicated to agronomy research in France, but also several universities, CNRS, that is basic research from France, and also the high school system linked to agronomy. So 8 institutions and 19 research groups in total, and the size of the area of the square is the number of groups in the different places. So of course I cannot go into the detail of all the labs in these places, but I think the inquiry will be given to everybody, so you will have all names associated to the different groups, and also more precisely the topics and things available in this lab. So here there are many scientific subjects, plants, microorganisms covered, and I put in bigger letters the ones that are the most relevant in number of groups and specificities. So there are specificities in soil microbiome diversity, microbial communities, and microbiome-mediated biocontrol of pathogens. There are groups, several interested in the impact of beneficial microbes on root development. Also on the evolution of beneficial plant microbe interactions, either mycorrhiza or bacteria. Genetics of the interaction, and signaling, and all these microbes are recognized as beneficial by the plant. For the different models we use, so there is a big community in France on the Medicago truncatula model legume, either to study rhizobia interaction and nodulation, but also mycorrhiza and even also PGPR interactions. There is also a big community, notably in Dijon, on P, and there are many others that are studied in more specific places. There is also cereals that are well recognized for the interaction with mycorrhiza, and a number of other crop of interest or more model plants. For the microorganisms, so there is also a large diversity that is covered, and mainly actinorhiza, rhizobia, and mycorrhizal fungi, but also more specific interaction also with pathogens. So the research scale that are covered, it's all from in vitro experiments and to till field trial, even though in the network the biggest interactions are at the level of, I would say, that the lab, and there are a bit less interaction between the labs working in vitro, in glasshouse, and in the field. So the facilities, so there is a collection of mycorrhizal strain in France. There is experimental farm for field trials. There is a growth chamber to study different parameters, but more specifically, that's not maybe available elsewhere, for light intensity and elevated CO2. There are different phenotyping platforms that can do high throughput phenotyping of plants' impact of beneficial interaction, notably on the root system. So there are some rhizotrons adapted to different plants, even crops. Phenotyping robot for root architecture, either in vitro or notably in vitro. And greenhouse equipped with load cells to follow drought response and more specific approaches. And for the tools, so there is strategies that are commonly used in many places, such as metabar coding and synthetic community, natural communities of microbes, and more specific traits also that I try to highlight, so notably in experimental evolution and in physiology, such as in studying systemic long-distance response to this beneficial interaction by grafting, split roots, and biocontrol assays. And the rest is more classical, I think. So thanks for your attention.
Next one is for Germany.
So, good morning, everyone. So first, before I start, it's quite new for me to see so many colleagues from so many countries dealing with the same topic. That's the reason why I'm here. I'm from Germany. I'm myself a fundamental and applied scientist leading a department on landscape functioning in the Leibniz Center, which is an institution. We are 500 people in total, and dealing with agriculture, transformation of agriculture to sustainability. And I'm teaming up with Caroline Schneider, and I guess I forgot an E in the end of the name. There she is. No, it's correct. She is a very well connected person from the application side, from a company that works now for several decades in the application of AMF fungi for crop production. So we were all busy, and what we have done here with the first survey is to give you an impression in which cities we think where our institutions and people responded that fit will benefit. Don't want to go in detail here, that you understand the situation in Germany. We have more than 120 publicly funded universities, institutions that deal with agricultural research. So I don't know if I covered everyone who's relevant. I know at least 2 or 3 that I would like to approach again and see if they respond. So there is a lot of potential, but how should I say, it's hard to grasp it. Here you see a word cloud from the people who have responded and delivered very unsystematically keywords, and you see what I like very much, that when we look for the host plants, when they are working on with microbiomes and beneficial microbiomes, we see here a lot of cereals, but also additionally tomato, some vegetables, grape wine, and of course some people are coming from this Arabidopsis field where all this research is fundamentally based in. So we have groups that, or sometimes the same groups that work from the plant level until field experiments. So for example, my institution can do both. And we have then expertise in the cultivation of these microbes, including our systems biology approaches. We can also work on the microbiome with several groups, metagenomics, these things. Plant system biology groups have also responded, and we have from the applied side groups that work on companies on the inoculation and application of microbes. Here I don't want to go in detail. You can read that. I guess you will share the slides afterwards, Benoit, right? And just want to highlight a bit my own institution, because we have restructured 6 years back completely. And 500 people, you can imagine that is some effort. And we are strong in European research when it comes to agricultural transformation, and you see we are starting new efforts in the more biology application part. And in the last 3 positions, you see names of people who are in my institution interested in the plant microbiome direction. There is one person more I will approach later. So that is what I can deliver here now in that moment. We have also found at least 2 companies, the one of Caroline and good collaboration partner, Christine Dieter, that will also contribute to our network here, but, and that is why I'm talking, I think we can extend this kind of network in the next few months by asking some people again and fostering forward. I also recognize that there is a parallel cost action on a very similar topic, and some of the German actors that I have in mind are more engaged there. So maybe we can somehow come together. And finally, I want to thank Benoit for the great organization. I know it's a lot of work to do these things, and I hope that we will have some fun in future and some productive moves forward. Thank you very much.
Next one will be for Greece.
Hello everyone, I'm very happy to be here. Thank you for the organization. So I'm going to give a brief overview of the results of the survey, people that correspond to the survey and give us further information for their work. So you can see we have institutes and research groups in 5 cities across Greece. With the red border, I have mentioned those that accept STSM, where you can find the research groups there that are willing to host people. And we summarize the results based on topics they work. Because the presentation will be shared with you. Also, the red people in each lab is again that it means that accepts STSM in their laboratory. And we have mentioned those people that have biocollections of microorganisms willing to work with them in collaborations. So we have 3 topics. The first one, it's about microbiology and molecular host microbe interactions. Here we have response from 3 labs. The first one is the Laboratory of Plant and Environmental Biotechnology of University of Thessaly, which the contact person is the MC member Kalliopi Papadopoulou, with which you can talk further if you would like. And they mainly work on the molecular mechanisms of multipartite and multitrophic interactions, how plant metabolites and small RNAs roll on it, have a role on it, and with synthetic communities. They work with model plants and with some crops like olive tree, tomato. They have specific microorganisms that test with a favor on Fusarium, Trichoderma, and AMF. In Athens and Agricultural University, we have the Laboratory of Plant Pathology, represented by Ioannis Triglis and Sotiris Jamos. They work more on plant microbe and pathogen interactions and use beneficial microbes as biocontrol and studying the whole microbiome assembly and how it is determined by the exudates and that is secreted by the plant during defense. Again, they have model plants and more applied crops, mainly tomato and eggplant, and they work with Pseudomonas traits and Bacillus, as well as Verticillium and Fusarium from pathogenic fungi. And in Heraklion, in Crete, we have the microbiology and molecular host microbe interaction group of Panos Aris that again look at the plant microbe interaction and the molecular mechanisms. And they are interested in PGPM that protect against biotic and abiotic stresses. They have a variety of brassicats and solanaceae plants interested, and they have a huge collection of bacterial endophytes. The other topic is the isolation, characterization, and application of PGPM microorganisms. We have response from 3 labs. The first one is the lab that I work in, the Soil Fertility and Biology Research Group in Agricultural University of Athens, where we focus on isolation from stressed environments, drought and salinity from the islands of Greece, and we try to find PGPM and AMF from their environments, and then we combine standard soil analysis, soil biochemical analysis, and molecular community analysis to understand how these all drivers help the plant to increase its fitness and to expand its realized needs under stress when mainly community-based microbia inocula are used. We have also a very big collection of isolates from stressed environments mainly, and we work with various crops, but also native vegetation of the Greek islands, called Phryganic vegetation. We have here from University of Thessaly and Laboratory of Agricultural Chemistry. They work with isolation and in vitro characterization and application of bacteria with plant growth promoting properties and how they help to the plant under abiotic stress. And they have also, they have experts on plant metabolomics. They have GCMS and LCMS units to do metabolomics, and they work mainly with vegetable plants. Yes, and from Hania Creek, again, we have the lab of phytopathology there, that they use PGPM microorganisms for bioprotection. And lastly, they work on nanomaterials and plant protection and how these nanoagrochemicals have targeted and non-targeted effect on the soil microbial diversity. Lastly, we had one response from a domological lab that they use mainly trichoderma, and they have a collection, a fungal collection, on biocontrol against pests. Yes, and that's all. Thank you.
Hello, I'm Peter Kau from Hungary. First of all, I want to apologize because for technical reason, I didn't use the provided template. Later, I can redesign it and provide the provided template for sharing. So, in Hungary, we have 3 members or interests in this cost action. I'm representing the Institute of Plant Biology, a Symbiosis group in the Biological Research Center at Szeged, which is in the southern part of Hungary. Agota Domonkos is representing here, she is in the audience, the Hungarian Agriculture and Life Sciences University at Gödöllő, which is close to Budapest. And we have a company who is interested in this action, the Biofuel small company, working on development of PGPR inoculant and these kind of okay, so just a few words about my home institute. So our main activity is the analysis of symbiotic nitrogen fixation. So we are mainly focusing on genetic analysis of nodule formation, infection, and rhizobia differentiation, and how the nodule functions. These are the main steps. So we are mainly interested in the specificity of the symbiotic nitrogen fixation. There are it involves the different steps at the beginning, from the recognition of the rhizobia and the bacterial differentiation. So. So now, as I mentioned, one of our main activities is the analysis of the molecular background of partner choice, mainly focusing on Medicago truncatula and different rhizobial strains. But we are planning to extend our research activity, how the interaction occurs between the symbiosis and the root-associated microorganism. We have different facilities in our home, in our home institute. So we have a nice phenotypic platform with the 3 units for bigger plants, larger than 1 meter height. For smaller plants, we have a growth chamber installed, compact system, and also the facility is running a root imaging system using Rhizothrone. And all of them is controlled by computer, and different visual scanning methods are available for this. There is another metabolomic service is also available in our institute, which can be relevant for this action, and they provide a full service from from designing the experiments, carrying out the experiments, and the data analysis. And so they provide a full support for this kind of experiments. As I mentioned, the other participant is the Hungarian University of Agriculture and Life Sciences. And they used to work on symbiotic nitrogen fixation, but following the restructuring in the back, they need to switch research profiles, so they started to work on root beneficial microorganisms. So there are two research groups. One of them is mainly focusing on fungal interactions with Solanaceous plants, how this interaction helps to cope the plants with abiotic stresses. And the other group, which is represented by Agota Domokos, they mainly focus on the soil microbiome, how it interplay with the plant development and mycorrhizal interaction. So there are a couple of facilities are also available here. And for example, field experiments can be feasible here or at the institute. And the last, the company which is interested in this action is focuses on development of microbiological and inoculants. This is not a very old company. They have a pipeline. Yeah, they have a full pipeline from isolation of microbiomes, characterization of the microbes, doing some fermentation steps, and finally they develop the soil inoculation. Yeah, thank you very much.
Next one will be Italy. Yeah, Israel will be after the break.
Hello everyone, I'm Mattias Pasquali from University of Milan. The presentation is together with Professor Lanfranco from University of Torino. As Germany, we have a huge country, so we decided to make a sort of cloud presentation. So as you see, first of all, an information how we did the survey. So we were trying to connect to people, but then also we use society, so scientific societies for getting answers. It seems from the answer that most of the society that was most active was mine, so the Plant Pathology Society. We also sent it to the Italian Botany Society, but most of the answers are possibly coming from the field of plant pathology. As you see, we have laboratories that replied from all over the country, and we go through the clouds presentation, so we have almost covering all plants, including forest plants, so everything. Most are horticultural crops, but also cereals, and some peculiar crops too. From the microorganisms of interest, we have a sort of predominance of, well, actually it's half and half bacteria and fungi. We have some, I mean, there's also some mistakes because there are also pathogens listed here, so that's possibly an issue, and people were replying to the survey. This is also something to consider when you send surveys to people, because they sometimes don't reply exactly to the question that you're posing, but it's okay. And anyway, we have expertise covering both bacteria and fungi. About the topics, we have a lot of biocontrol activities, as well as PGP activities. We have mycorrhizal studies and the use of bacteria also for rhizal remediation, so using bacteria for purification of the soil. For what concerns the tools and approaches, the statistics has no value because, I mean, it's not really comprehensive of the country, but they are covering from lab activities to field, and at least 30% of the lab have also activities towards field applications. as you see, you have all mixer tools, molecular biology, some specific genetic activity, as well as physiological analysis. And that's the, let's say, very partial overview of Italian situation. So maybe for the final publication, I think, it would be worth having some extra requests from people that we know are working that didn't reply so far. Okay, that's it.
We're going to have a last one before break and then continue after the break. So the last one will be Latvia.
Hello, dear colleagues. We have prepared a brief summary which consists of 3 independent parts and which will be presented by Olga, Zygmunds and Darta during these 5 minutes. I represent the University of Latvia Faculty of Biology, and one of the most recent projects was focused on the optimization of air biofilters, or botanical filters or green walls, the same, for purification of air using the formaldehyde as a model contaminant in a hydroponic system. So in this project, which we conducted in cooperation with industrial partner, LAFEvents, and our colleagues from our faculty, plant physiologists, headed by Professor Gedert Jevinš, we compared the effect of bioaugmentation, the concentration of formaldehyde, plant species. We compared the efficiency of this air biofiltration process. So we collaborate also with the Latvian Biomedical Research and Study Center. They are responsible for the molecular tools, so we can compare the shift of metagenome during our treatments. And we control this process. We test this process using a broad range of methodical approach, biochemical and microbiological. We measure this enzymatic activity of biofilm, which is attached to the ceramic expanded clay pellets, and we test the changes occurred on the surface of this substrate of hydroponic system. Also, how plant biomass reacts to these treatment types. Another reaction, this is soil microbiology, soil health, and mostly we are focusing on the biodegradation processes, bioremediation technologies, including phytoremediation. And here we apply also a broad range of treatments, and particularly this, we use the biochar in the broad context. And here just one sample how we tested the effect of biochar amended with Trichoderma viride on the growth of corn. And overall, now we have a current project related to the biodegradation of plastics. And we would be very interested to collaborate with you who are working also with plastic biodegradation, so to study this very complex process in relations of microorganisms with biodegraders and higher plants. And no data, please.
Thank you, Olga, for inviting me. I'm from Forest Research Institutes here in Latvia, so the topic is a bit different, but I think there are a lot of things we can interact. We have focused mainly on forest pathogens, but here I put some slides about ectomycorrhizal research we have done on pine and spruce. First topic we were concentrated was different fertilizers applied to the system, like natural system. We are always studying in natural forest, either under greenhouse greenhouse conditions, and here this study was about applying different fertilizers and how mycorrhizal system responds on it based on morphotype analysis, and afterwards IDS Sanger sequencing of those fungi present in the system. And then other topic was more about former land use, what impact it has on ectomycorrhizal and soil fungal community. There we use NGS sequencing with PacBio, and I think in one experiment it was also Illumina. And so different land use and different soil preparation, so either with stamp removal and not, so what response it has to the fungal community. And there we have close collaboration with Estonian colleagues. They mentioned already with those labs, and I think we can continue it also in this framework. And then also in Silo we have one project in common with Sigmund, so I think he can continue.
Good morning, everybody. Very briefly, I wanted to use the opportunity to introduce my emerging research group, currently based at Latvian Biomedical Research and Studies Center. Apologize for the few font missing here. Yep. So we are actually addressing this hypothesis. We're interested in understanding how arbuscular mycorrhizal fungi could serve as a medium that connects two plants together, and whether they transmit signals between these plants that could be functionally useful for the receiver plants in defending themselves against pathogens. So we are using transcriptomics and targeted metabolomics and a lot of pathogen assays. We are using model plants such as carrot hairy root systems and Medicago plants, and as Darta mentioned, collaborating with our Forestry Research Institute to use hybrid aspen and birch as models also for mycorrhizal colonization. And our preliminary data already suggest that arbuscular mycorrhiza could play a role in different plant responses, including volatile production by receiver plants, and that is also associated with different pathogen response in receiver plants, in this case Medicago. I would very much like to, if you're interested, to discuss any aspect of this work with you today, and I'm very much looking forward to also finding new potential collaborators and some joint grant applications if anybody would be interested in collaborations. Thank you very much.
So it's almost 10:30, so it's time for break now. For those who have not yet sent me the presentation, you can just come and upload it. And we have a half an hour break, I think.